Okay. Thank you for the introduction, and thank you to the organizers for the opportunity to present my work here at ISMB. And today I'm going to talk about some of my work from my thesis, master thesis. So domains are sequence fragments that encode protein modules with the distinct functions and structures. The insulin receptor, as shown here, is a multi-domain protein. This schematic shows the domains as colored boxes, as colored shapes. Viewing the 3D structure from insulin receptors with the domain colored as in the schematics, we can see how domains corresponding to structure unions. And as observed in the previous literature, protein domains vary greatly in their promiscuity, the propensity to form different combinations with other domains in the same protein. For example, this figure from the literature shows the SH2 domain in gray with many different domains it combines with. Will some domain appear in many different combinations? Others are observed only in one or very few combinations. So what determines domain promiscuity remains an open question. Domain may be promiscuous because they facilitate the evolution of novel protein functions. For example, the linkage binding domains can modulate the function of many different catalytic domains. Or domain may be promiscuous because they are mobile. So mobile domain can be easily inserted into and copied into new contexts and fold independently in many contexts. So here I focus on association between domain promiscuity and mobility. And I consider two different hypotheses. The first one is that domain mobility is a significant deliverer of domain promiscuity. And second, we consider the hypothesis that domain length is linked to domain mobility, which is inspired by the idea proposed in previous works that smaller domains are more likely to be mobile and found in multi-domain proteins. And prior studies have probed those questions by inferring events on a tree using different parsimony models like Fitch and Dolo parsimonies. And here we investigated those hypotheses using domain gain and loss rates as a proxy for domain mobility. While parsimony models gave a lower bound on the number of events happened, rates provides a more dynamic assessment of mobility. So how did we go about investigating those hypotheses? And if given a set of domain architectures and a species tree, we can infer the domain family event rates, and we can also obtain promiscuity values and domain lengths for those domain families. And then we can compare domain event rates with domain promiscuity to investigate the relationship between mobility and promiscuity. So in this work, we considered the set of domain architectures encoded in the genome of 19 Metazoa species and the 2 unicellular outgroups. The domain architectures were downloaded from the superfamily database, and the dataset contains 1283 distinct domain families. And now with the domain family dataset available, the first step is to infer the domain family event rates. And here we infer the domain family event rates using a probabilistic birthday scheme model implemented in the software account. So given a species tree and the domain family abundance in present-day species as the inputs, this model infers the rate parameters that best explain family sizes in present-day species using a maximum likelihood estimation. And in this model, the rates were allowed to vary across both species lineages and the domain families. And here, we will focus on family-specific rates only. On the right-hand side, you can see we show the overall distribution of inferred domain family gain, loss, and duplication rates. And those are the rates we will be used in subsequential analysis. And the second step is to quantify promiscuity. I'm going to show you several different promiscuity measures that have been used or introduced in previous literature. Then I will use correlation analysis to see whether or not those measures capture the same information. And I will also test how domain length correlated with those promiscuity measures. So many different promiscuity measures have been proposed earlier. And here we focused on the number of different domains that co-occur with this domain. In this tool example here, different shapes represent different domains. For example, domain B in circle co-occur with 4 different domains, the A, C, and E, F domains. And we also have the number of bigram or pairs that contains the domain, which represents more localized context. In the same example, the domain B forms 3 different pairs, the AB, BC, and BE pairs. Another measure we have is the number of trigrams or triplets that contains the domain. And here, domain B only appear in the middle of 1 triplet. So we know that domain promiscuity is bounded by domain abundance. That means the more copy of a domain you have, the more neighbors it can have. So therefore, we also considered a measure of domain abundance, the number of times the domain appears. And in addition, we also created 3 normalized measures. Dividing by the domain abundance gives an assessment of promiscuity of a domain, giving the maximum number of neighbors it could have. And other authors have proposed various measures that account for domain abundance. For example, the weighted bigram frequency index. This is a measure of relative entropy, where the bigram frequency is normalized by domain frequency in the second term. And since the sets of domains has also been suggested to affect mobility, so we also considered domain amino acid sequence length for each domain. So to assess the agreement between those various measures, we calculated pairwise Pearson correlation coefficients. And we can see the correlation matrix shows those promiscuity measures form 2 different groups. And we observe that all raw promiscuity measures are strongly correlated with each other and with abundance. The normalized promiscuity measures are not correlated with the raw promiscuity, which suggests that the raw promiscuity measures are evidence of abundance rather than an elevated propensity for forming different combinations. Also, for the weighted bigram frequency index, this measure was designed to account for abundance, but in fact correlates with the raw promiscuity measures. And lastly, we observe that domain size is not correlated with either promiscuity or abundance. So now that we have the inferred domain family event rates and the promiscuity values, we're ready to ask whether domain promiscuity is associated with mobility. For that, we use the linear regression model to determine whether domain event rates are predictive of domain abundance and domain promiscuity, and the significance of those relationships. The same analyses are repeated for domain length. So here we plotted the coefficient values for each event rate from regression model. Each bar color is associated with a different measure, and they are grouped by event. And we can see that the abundance and promiscuity are positively correlated with skin and duplication rates. And they are negatively correlated with loss rates. And meanwhile, contrary to suggestions in the previous literature, our regression model results suggest that there is not enough supporting evidence for the hypothesis that smaller domains are more mobile. Since we know that now promiscuity is correlated with mobility, abundance is also correlated with mobility. And moreover, we already saw that normalizing by abundance had a huge effect on promiscuity measures. And promiscuity is bounded by abundance. So we wonder whether the significant correlation we observed is a byproduct of abundance. So to answer that question, we performed linear regression using a more complex model in which abundance is included as an independent variable. And here we will use the result for bigram frequency as an example. We found that both ASA and BSA values suggest that the more complex model, Model 2, has better explanatory power. And this figure compares the coefficient value obtained for the two models. The coefficient values for the original model are shown in red, and the result for the new model, which includes the abundance as an independent variable, is shown in blue. When abundance is included in the model, coefficient values show that promiscuity is highly dependent on abundance. And note that there is no abundance coefficient for model 1 because it's not included in that model. And promiscuity shows a weaker and less significant correlation with skin and duplication rates. The height of the blue bars is reduced compared to the red bars. Moreover, we found that promiscuity is not correlated with loss rate anymore. The blue bar here is almost 0. So in summary, our preliminary results shows that mobility is definitely a strong contributor to domain abundance. And after controlling for abundance, the contribution of mobility to domain promiscuity is weak. And also, we observe no relationship between domain length and domain promiscuity. So I would like to thank Dr. Danny Duran, Dr. Maureen Stolzer, and all my previous and current lab members. And thank you for your time and interest. I will be happy to take questions.
Thank you very much, Yuting. Very interesting presentation as well. Nice addition to the previous presentation. Do we have questions in the room for this presentation? No? Okay, so I will go with my questions before question in the Q&A maybe. So I was wondering in this analysis if you have taken into account the fact that you may have some domains that are duplicated, that are there several times. So in the promiscuity, are you looking also at the promiscuity between a domain and its copy, for instance?
Yes. So for example, for the race data, we use the abundance of domain family in present-day species as an input, and those copy numbers could be resulting from like duplication of a domain, a tandem of the domain, and in both gene level or in domain level. So those are all considered behind the race. And for promiscuity variables, we follow the practice from previous literature that using, actually I can show us, sorry. Sorry. Yes, here. So for promiscuity measure, we actually collapsed our abundance value for tandem array. For example, here you have 3 different domain A in this example, and the abundance is 3. But if you have like a tandem array here, we count it also as 3.